Hey guys, in this video we're going to walk through a kidney model where we can see the macroanatomy. We can see a section through the side of the kidney, and we can also zoom all the way in to the renal corpuscle over here. Let's start with the macroanatomy, the gross anatomy. This is the entire kidney. All the way on the surface we can see the fibrous capsule. Okay, so it has that like kidney bean color here. If we section through the kidney, what we can see is that the organ is broken down into the outer layer, which is the renal cortex, and this entire middle layer here, which is called the renal medulla. Note that the renal medulla is broken into sections, these right here, which are called renal pyramids. So the structure is the renal pyramid, but the region of the organ is the renal medulla. Also note that the renal pyramids are separated by cortical tissue that dips down in between adjacent renal pyramids and divides them. And so this structure here is called the renal column. Okay, so when in doubt, say renal. Okay, as we're going to see elsewhere on this model, each individual renal pyramid is going to produce urine. And that kind of happens here over this corticomedullary boundary. Nephrons drain into collecting ducts, which we'll look at in a moment, and ultimately lots and lots of collecting ducts drain collectively into this funnel shape right here. A single funnel-shaped structure that drains a single renal pyramid is called a minor calyx. So here is a minor calyx, here is a minor calyx, here, etc. Where multiple minor calyces join together, this is called a major calyx. Note that these circles here, these little indentations, what these are is essentially showing us that this is a three-dimensional structure, and there are other minor calyces which are going into the model. Okay, so this is just a single plane, but in fact there are calyces and pyramids pointed in this direction, and that direction as well. So again, minor calyx, minor calyx, minor calyx, together a major calyx. And where multiple major calyces come together, okay, and just a note on that terminology, a calyx is singular, calyces is plural. Where multiple major calyces come together, there is a structure called the renal pelvis. And so this pelvis essentially collects urine from the entire organ and allows it to drain out a single tube here, which is called a ureter or a ureter. Okay? The ureter exits from the medial indentation on each kidney, called a renal hilum. Okay? Also entering and exiting from this same indentation is the renal artery and the renal vein. Ultimately, the ureter is going to deliver urine down to the urinary bladder before it exits out the urethra. Okay. Next, let us take a section through the kidney. Right, up here, this is the fibrous capsule. This section here, all the way down to these blood vessels, this is the renal cortex. And so down here, this is the renal medulla, specifically part of the renal pyramid. As we can see, there are a series of tubules which kind of wiggle throughout the cortex, and some of them dip way down into the renal medulla. Let's just walk through, one of these tubules. Okay, so the first thing we see here is Bowman's capsule, or the glomerular capsule. If we were to take a section through that, we would see over here, right, this yellow sphere, just like this. There is a capillary bed called the glomerulus inside, surrounded by Bowman's capsule, or the glomerular capsule. Okay? This is where filtrate is formed, and ultimately the filtrate from Bowman's capsule is going to flow through this first squiggly portion right here. And so we call this the proximal because it is near Bowman's capsule, convoluted because it squiggles, so it's convoluted, it's not in a straight line, tubule. Okay, so this is the proximal convoluted tubule. Okay, when the PCT, or proximal convoluted tubule, straightens out, the filtrate dips down and descends either closer to the medulla or actually down into the medulla as we see here. This is called the descending limb. Filtrate does an about face and starts to come back up again. This is the ascending limb. And together, these two are referred to as the loop of Henle, Henle's loop, or the Nephron loop. Okay, so filtrate has ascended back up, and it enters into yet another squiggly part of the tube. Okay? We know that squiggly parts of the tube are called convoluted tubules. This one is proximal, this one is distal. Note that the distal convoluted tubule passes right by Bowman's capsule. And essentially we are monitoring the end result of this filtration, secretion, and absorption process so that we can modify the next go around for the next set of filtrate. Okay? Note that not every PCT is off to the right here, not every DCT is off to the left. In fact, in reality, these two might kind of wiggle together here, right? They're woven around each other. Here we have the PCT on the left and the DCT on the right. Okay, so what makes a proximal convoluted tubule proximal is that this is the first structure that actually receives filtrate from Bowman's capsule here. Distal is ultimately going to drain the filtrate into this long tube right here. Right, this is the collecting duct, and this is ultimately what drains the filtrate, now we can call it urine, down out of the renal medulla, okay, into the minor calyx. Okay. A couple more notes here. Some of these, well, actually, before I get there, the PCT on this particular model is always yellow. The DCT is always white. Also, you can figure out DCT versus PCT just by looking for where the filtrate would flow and just kind of following it around. Similarly, once you find the PCT, you know which one is the descending limb and which one is the ascending limb, regardless of which side they're oriented on. Okay. Also, the collecting duct here, we can see has a lot of different branches. Right? Also, each collecting duct collects filtrate from multiple distal convoluted tubules. Okay. All right, over here, we can see Bowman's capsule. We can see what is the afferent and efferent arteriole, okay, taking blood into Bowman's capsule, into the glomerular capillaries, and draining some of that blood out. Next, we can see these other capillaries, which are kind of wrapping themselves around this entire structure here. Down here, around the loop of Henle, these capillaries are called the vasa recta, literally the straight vessels. And up here in the renal cortex, what we see is the peritubular capillaries. Okay, so cortical capillaries are peritubular. These down here in the medulla are the vasa recta. Okay, just a couple more notes before we zoom in just a little bit farther. Again, here's the cortex, here's the medulla. Note that this particular nephron does not actually extend down into the medulla. And so this would be called a cortical nephron, whereas this nephron right here is called the juxtamedullary nephron. Okay, it is next to, or actually within, the medulla. And so the loop of Henle dips way down into the medulla here, and that's important because this is concentrating the urine quite a lot, given this very long structure. Okay. So, once again, that side, we were looking essentially at this boundary between the cortex and the medulla, right? The structure of the tubule kind of wiggled around up here, dipped down into the medulla, and then finally drained its contents into the collecting duct, minor calyx, major calyx, renal pelvis, and finally the ureter. All right, one more structure. This here is what is called the renal corpuscle. All right, together we have the glomerular capillary, so that's the actual capillary bed, surrounded by Bowman's capsule or the glomerular capsule. All right, note that the parietal wall of Bowman's capsule has these simple squamous epithelial tissue, or is made of simple squamous epithelial tissue. Okay, the visceral layer is actually made of what are called podocytes. And so if you look really closely, you can see that these cells have a lot of little finger-like projections, which actually weave together to create filtration slits. Okay, so the entire structure here is called the renal corpuscle. Just the blood is the glomerulus. Just the capsule is Bowman's capsule. Together, renal corpuscle. Okay, so once the filtrate is generated here, it is going to drain out here via the proximal convoluted tubule. Okay. Coming up to the top here, what we can see is another portion of what is called the renal tubule. Okay, so this is where the filtrate would be going, and this happens to be the distal convoluted tubule. So we're essentially monitoring what is happening in the renal tubule by looking at the end product. Okay, and so this is going to determine the amount of blood that's flowing in, it's going to determine if hormones are going to be released, etc. Okay. On that note, this model also shows us the incoming blood. Okay, so this here is the afferent or afferent arterial. Or really high blood pressure or glomerular capsular pressure in here. Filtrate is formed over here, and any of the blood contents that don't cross over this filtration membrane and finally into the rest of the renal tubule are going to exit the glomerular capillaries via this vessel right here. This is the efferent or efferent arterial. Okay. Note that there's a lot of smooth muscle around both the efferent and afferent arterioles, and essentially we have very fine control over how much blood is coming in versus how much blood is leaving. And that's really important to regulating our blood pressure, both within the glomeruli as well as within the entire body. Okay. There also is another series of specialized cells. We can see here that there are some cells that are columnar as opposed to cuboidal. Right? These cuboidal cells are great at filtration. Right? So they are secreting and absorbing and doing all the things that the DCT does. But these cells, they have another function. These cells are monitoring particularly the salt content within the DCT. Okay, so these cells are called the macula densa cells. Okay, and again, they are regulating or monitoring what is going on in the filtrate here. Okay, there are also specialized cells over here in the afferent arterial. These are called juxtaglomerular or granular cells, and these are responsible for monitoring stretch in the afferent arterial, therefore monitoring blood pressure, and under conditions of low blood pressure, these juxtaglomerular cells, JG cells, can actually release renin or renin into the blood to ultimately stimulate the RAAS, right, to bring up systemic blood pressure in a major way. Okay. Final set of cells here. In between the afferent and efferent arterioles, there are what are called extraglomerular mesangial cells. Okay, so these are also regulating kind of what is happening at the end, what is happening at the beginning, and they play a role in talking to the afferent and efferent arterioles to ultimately determine glomerular filtration pressure, right? How much blood is coming in here, how much pressure, and therefore how much filtrate is being formed down here. Together, the juxtaglomerular cells, extraglomerular mesangial cells, and the macula densa cells are called the juxtaglomerular apparatus. So right next to the glomerulus, we have an apparatus that is monitoring blood pressure, monitoring filtrate, and that is going to determine glomerular filtration rate down here, and ultimately how much urine versus blood we are going to use or produce at the end of this filtration process. Okay? So thank you guys so much for watching. We looked at, we zoomed in really far to the renal corpuscle. We looked at the gross anatomy of the entire kidney. And then we also zoomed into the renal cortex and the renal medulla, looking at this structure right here. Anywhere that the filtrate is touching, is flowing through, that's called the renal tubule, collectively with the blood as well. So